Hello everyone, and welcome to today's webinar, Advancing Cellular Ultrastructure Research with Freeze Fracture and Freeze Etching. I'm Alexis Corrales of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Leica Microsystems. To learn more, please visit their website at leicamicrosystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window located at the bottom center of the screen to obtain your credits. I'd like to now welcome our two speakers for today. Evan Krystofiak, EM Staff Scientist at Vanderbilt University, Cell Imaging Shared Resource Core Facility, and Seth Villarreal, Advanced Workflow Specialist, Nanotechnology at Leica Microsystems. Gentlemen, welcome. Evan, you may now begin your presentation.
Thank you for that introduction. Today I'd like to tell you about a technique near and dear to my heart, freeze fracture and freeze etching. This talk will be broken down into three parts. First part is a general overview of freeze fracture and freeze etching, going over the general principles when you would use freeze fracture, when you would use freeze etching, and the workflow involved in producing these replicas. Second part will have some real-world applications of studies that I've done that have used either freeze fracture or freeze etch. These are cloud and strand morphology, so looking at Ti-Junction strands by freeze fracture, and the enteric glycocalyx architecture, which we use extensive freeze etching for. And then the third part will concern optimizing the replica images themselves. So there'll be some general guidelines on how to produce very high-quality replicas. And the second part will concern a optimization technique that I developed to improve the biological fidelity of replicas themselves. So let's start with freeze fracture. Freeze fracture is characterized by physically fracturing frozen samples along their lipid bilayers. And this will produce images of either the extracellular or cytoplasmic face of the leaflets, including the membrane proteins. So I've got a couple examples to show of what these kind of images look like from freeze fracture. So at the top, we have a cell that's been fractured, and we can actually see the interior of the cell here. So we have a lot of cytoplasm, and there's a lot of vesicle trafficking going on. And then here, we actually have the cytoplasmic leaflet of the cell with a lot of membrane proteins in it, including a very extensive tight junction, which is located here. Down here in this middle image, we actually have a much higher magnification image of what these tight junction strands actually look like. So this is, again, the cytoplasmic leaflet, and we can see these tight junction strands throughout it. And down here is another example. This is a red blood cell leaflet showing how many proteins are actually present within the red blood cell membrane. Now, freeze fracture is very useful for biological details of cells, but it can also be used to characterize other structures such as exosomes and liposomes. The advantage of using a freeze fracture technique for these kind of samples is they will be well preserved, so you won't have the same kind of artifacts that you would observe in a dried sample like a negative stain. So let's go over how freeze fracture is actually done in principle. You have a frozen sample that is in a freeze fracture apparatus on a cryo stage. Cryo stage is typically kept around -110, but that temperature can vary depending on what your exact goals are. And we have two deposition sources, a carbon source and a metal shadowing source, and a cold microtome blade that is kept as cold as possible. Typically, that's around -180, but sometimes you can do a little bit better. So we use that blade to actually fracture the cell, hopefully along the lipid bilayers. Once the cell has been fractured, we want to kind of rapidly produce a metal shadow from an angle. For freeze fracture, we typically do unidirectional shadowing at a 45-degree angle. So unidirectional means there's no rotation in the stage. So we're angling just from one side. And this will produce a really nice shadowing effect that is easy to interpret most of the time. As soon as that platinum has been deposited, we want to stabilize the whole replica with a layer of carbon. And this is done from the top. If you do not stabilize the replica with carbon, it would actually just fall apart because it wouldn't have any cohesion. So the carbon is deposited typically with rotation to make sure you get a nice even coating. Let's go on to freeze etching. So freeze etching is a similar technique to freeze fracture, but it's mostly used to look at things outside the membrane or maybe attached to the membrane, but not within the leaflet itself. So freeze etching is characterized by sublimating water off a frozen sample and then creating a replica of that surface. It often still includes fracture to make sure that we've cut into the tissue or the cells to the correct depth. But the technique is really characterized by looking at these kind of external or internal structures. So I have a couple examples here. This top image is the mouse glycocalyx on the small intestine. So these are microvilli. This is glycocalyx, which is this extensive meshwork structure that sticks out. Here we have a variant of freeze etching called unroofing, cell unroofing. And what we're looking at here is actually the inside of the membrane from the cell perspective. This is a clathrin-coated pit visualized from the inside. And down here we have, this is the luminal surface of bladder epithelium showing this kind of crystalline protein array that exists on the surface called uroplakin proteins. So this can also be used, this technique, freeze etching, can also be used very extensively for exosomes, liposomes. Again, this would be looking at the surface of these instead of looking inside, or even hydrogels. The glycocalyx is sort of an example of a naturally produced hydrogel that exists in the intestine. So hydrogels often suffer from dehydration artifacts. Typically, if you want to look at it by SEM or TEM, you're going to have to go remove the water. When you do this by a chemical dehydration, such as the details, and this can be problematic. So let's go over how you do freeze etching. It's a somewhat similar workflow. So we start with the same kind of idea. You have your cell, it's at -110. We have our cold blade. The E-beam for the platinum shadow is now at a different angle, and we'll go over that in a minute. So we're going to fracture our sample. You can actually fracture above your sample in this case if you would like. I typically aim to hit a little bit on the sample. And rather than have the microtome immediately clear the sample to do the metal shadowing, in this case we're going to leave the cold blade over the sample and then warm the sample up just a little bit to -100 C. Now the temperature differential between the sample and the cold blade along with the vacuum is going to cause water to leave the sample and get attracted to the knife blade. And depending on how long you do this, you can get either a very shallow etch, so that would be maybe 5 minutes, to very deep etching, which can go up to 30 minutes. So once the water is sublimated off the sample, we'll move the blade, and then we will deposit the metal at an angle. Typically, I like to do this with rotary shadowing, so the sample's rotating, and we will do this at a lower angle. So with rotary shadowing, it's often ideal to reduce this angle to get a little bit better detail, especially when going for details of these extracellular structures. So once the platinum has been deposited, we do carbon stabilization again, and this is just to hold the replica together. And now we have a complete sample. Now what do you do when you have this done? Well, here's the whole workflow. So to start with, you need to have a frozen sample, and it needs to be in vitreous ice. You cannot do freeze fracture, freeze etch on a sample with ice crystals. It will look extremely poor. So your options are typically high pressure freezing or slam freezing. Both have their advantages and disadvantages. High pressure freezing will work very well and freeze your sample all the way through, often with vitreous ice, but doesn't work on all samples. Slam freezing, on the other hand, is pretty consistent, but will only freeze about 10 microns of depth, so not nearly the depth you can get with a high pressure freezer. Now, you can freeze in other media, provided that you get, or using other machines, provided you get actual vitreous ice. One way of doing this without this equipment is to cryoprotect, so you can fix samples, infiltrate them with cryoprotection. For freeze fracture, and glycerol works really well. And then you can plunge freeze in something like liquid ethane. That's a little bit more accessible. But you lose a lot of the advantages of freezing live and getting the biological context of your tissue. So once you have your frozen samples, you need to transfer them to the freeze fracture apparatus, and you need to maintain them in their vitreous state without contamination. Then we go through the process we just described, so fracture or etch, and then depositing the metals. And then we have a couple options. So my preferred option is to image on the TEM because it's higher resolution. But before you can do that, you need to remove the cells and tissue from the sample. So we actually only want to image that metal and carbon film because we won't be able to image throughout the tissue itself. So typically this is done by floating the whole sample on bleach for quite a while until all the sample is removed. You can also float on hydrofluoric acid if your sample's on a cover slip to dissolve the glass, and then after some washes, float on bleach. So that's how to get to the TEM. Now, if you have a cryo-SEM, you can actually do a direct transfer between the freeze fracture apparatus and the cryo-SEM. And the advantage to this is that your whole sample will definitely remain intact and you'll have good biological context for where you're imaging. The disadvantage is you won't have the same resolution you would have in the TEM. All right, so let's move on to the second part. These are the real-world applications that some studies have done. We're going to start with tight junctions. So tight junctions are a cellular feature that exists between, or exists in epithelial and endothelial cells. It is a network of sealing strands right at the apical surface that serves to moderate the ion flow between the apical and basal side of the cell. And this is essential for a lot of different things, including blood-brain barrier, proper intestine function, proper hearing. There's quite a few examples. The main protein involved in tight junctions are claudins, and these are what actually physically form these strands. Now, these strands can be observed by a variety of different techniques. We can use confocal with a claudin that's tagged with the GFP, for example, or you could antibody label. And we get images like this showing that there is a tight junction here, but we don't have a lot of details beyond that. You can also look at them by TEM. This is two enterocytes in the small intestine. This is our apical surface right here, and we can see the tight junction. But again, we're not getting a lot of details about what that structure actually looks like. So let's look at it by freeze fracture. So this is images of the claudin 15 strand fracture. So in these images, we can see a lot more details than we could by confocal or conventional TEM. In this case, we're seeing that there are multiple strands in a tight junction. They are curved. They can either have a gentle curve, shown here, or these really kind of quick deflections, and they can also branch. So these are all details that would not be observable without freeze fracture. So in this study, shown here, we did a lot of characterization of how these strands can bend, flex, and branch, and we're able to characterize sort of some of how that works in the strands. Now, also, this figure shows a very important thing, which is the difference between the leaflets. So this is the P face. This is the cytoplasmic face of the leaflet. And this is where the proteins tend to segregate to during a fracture. So all the strands are in the P face. The E face, on the other hand, which is the extracellular face, will tend to have these grooves in it. And this is where the proteins were, but they're no longer present. I find the P face easier to analyze than the E face, but both are valid. So one of the major questions about Ti-Junctions is sort of how they are built. So in 2014, Suzuki et al. Published in Science the first structure of a Claudin monomer. This was the Claudin 15 monomer. And this set off a whole series of studies in how this monomer is assembled into this strand-like structure. So this same group proposed a structure that was rather complicated between that shows how the Claudins are assembled both between cells and in a single cell. So the tight junctions themselves are actually always between two cells, so it can get a little confusing to image. Here's cell 1 membrane, cell 2 membrane, and the Claudins themselves have a trans interaction. So trans means transcellular interaction. Now, if we look at the same structure kind of from a top view down, instead, we can see what the structure looks like in their model. And this actually has two different interactions that we call cis interactions because they're in the same membrane. So we have a cis interaction along the strand. You can kind of imagine the strand growing this way. And then a second interaction that occurs across the strand. So we were very interested in validating this interface here that was proposed by this group. So we started by looking at the crystal structure and what residues were proposed to interact in the previous study. And that was the methionine 68 in Claudin 15 with the phenylalanine 146, 147. Now, this 146, 147 are well known to be important, and if you mutate those, strands do not form. However, when we examined this methionine, we found that actually when you mutate it to either alanine or glutamic acid, it doesn't really make a difference in strand formation. So we can see by confocal that we have tight junctions forming in these cells. And then when we do the freeze fracture on these cells, we can see that the strands themselves look pretty similar to the wild type. So this suggests that this particular residue is not that important. So this sent us back to kind of look at what might be going on here and what this interface might also kind of imply in the flexibility we saw in the strands. So we did some analysis on the residues involved in the interface here, and we found that there are actually some very conserved residues among all the different Claudins, suggesting they're probably important in the interface. We also found that if you rotate this interface a little bit from the crystal structure, that you get a kind of a more energetically favorable interaction. And this might be a good mechanism for how the strands actually bend. So from our analysis, we determined that the serine 67, right next to the methionine 68, is probably pretty important because it's forming hydrogen bonds, along with this glutamic 157 on the adjacent Claudin. So we set out to kind of do the same type of mutagenesis to see if these residues were important. And actually, they all formed, no matter what mutation we made, they all formed tight junctions on the confocal level. So if we left it at that, it would be kind of a really boring story that we did these mutations, we analyzed them by confocal, and we weren't able to see very much. However, in freeze fracture, the strand morphology changes quite a bit. So here's our wild type strands again. So this is P face. Up here we have a little bit of E face just to show you the grooves. If we mutate that serine to an alanine, we end up going from these nice continuous strands in the P phase to kind of a broken strand morphology that mixes between the P phase and E phase. Similar to the glutamic acid, when we mutate that, we also get a broken morphology. And this is probably the most interesting part of the study. We can actually swap these two residues, so the serine to glutamic acid to serine, and that will actually form a normal-looking strand network, which is really kind of cool. If you do the single mutations, they still have the broken morphology. So this suggests this is sort of a rescue by maintaining the same interaction, just swapping which Claudin is doing the interaction with the other. So this, the fact that they still form junctions, but they're disrupted, suggests that this is a cis interface that's important, but probably not the only cis interface that exists. It's probably a second one that's allowing the strands to at least partially form that we can detect by tight junction or by freeze fracture or by confocal. That's probably that second cis interaction that we saw across the strand. We'll revisit that in a little bit. Let's move on to the second study. This is concerning the enteroglycocalyx. So the glycocalyx is a glycoprotein covering that surrounds or is on the apical surface of epithelial and endothelial cells, and also can coat bacteria. And in all three cases, it often serves to act as sort of a protective barrier to these cells from their outside environment. Now, the enterocyte glycocalyx is primarily composed of mucins. So we use that term kind of interchangeably, glycocalyx and mucin. But these things are actually notoriously difficult to image. So we can image them by confocal again, and this is done typically by doing immunogold labeling. So we can label with wheat germ agglutinin, and this sort of just labels the sugars in the mucins. Or we can actually use a mucin antibody, in this case, mucin 17. And we can get some details. We can detect that these structures exist on top of microvilli, which are labeled here with phalloidin. But we can't really get an idea of what this structure actually looks like. We can't even really get dimensions on it. Now, if we try and look for this glycocalyx by an EM method, you actually have a lot of difficulty. So here's SEM of an enterocyte kind of fractured in half. This is not freeze fracture, this is just SEM fracture. And if we look on top of the microvilli, we don't see much of anything. Similarly, if we look at pre-substituted enterocytes and TEM, we get this haze above the microvilli, but no definitive structure, nothing certain. So let's see what this looks like by freeze-etch. When we freeze-etch this structure, we have something quite striking. So we have our enterocyte, microvilli, and then on top of that, we have this kind of huge glycocalyx structure that sits on top of all these microvilli. And if you look, it's almost the same size as the microvilli cells. Let's compare that again to the SEM image, which doesn't show anything. If we were to go by the SEM image, you would assume that the microvilli are actually bare, when in fact this large structure exists on top of the microvilli. What can we do with this kind of imaging? We can actually learn a lot about how this glycocalyx functions with the enterocyte. So just from this image, we were able to determine that there are several extracellular vesicles that appear kind of in the glycocalyx themselves. They may be going or coming, we don't know, but they are sort of trapped in here for now. We can see that where the mucins leave the microvilli is a different location than where the cadherent, protocadherent links are that bind the microvilli together. So the mucins are more apical, really just from the tip where they kind of fan out and interact with each other. We're also able to look at the mucin glycocalyx network and kind of characterize some of its sort of filter functions by looking at the mesh size. So this is sort of the pores that form in between this kind of complicated strand structure. And this is where you could get a comparison to something like a hydrogel. And we also were able to see how this mesh structure actually kind of forms. So the strands or the filaments of the glycocalyx kind of merge together and branch away at various points the whole structure to create these kind of void openings that we call the mesh. This occurs in just multiple different ways. We can also see that the filaments themselves actually have different widths, and this suggests there's multiple mucins involved, which isn't too surprising. And then at the top is a really kind of astounding feature. So at the very top of the glycocalyx, we have what we call the termini. So these are the ends of the filaments. And one of the most striking features of the termini is that they all kind of end at the exact same plane. And if we look at them, each one has a little dot, and they're actually hexagonally packed from each other. So we can see that here with just our eyes, but we can also do a Fourier Transform and kind of show that in more detail. So we were able to show that this termini all basically have the same nearest neighbor distance, and they're packed very carefully, suggesting that while we have kind of chaos below with the strands coming together and coming apart, there's a lot of order actually in the system itself. The other interesting thing with these termini is they are very resistant to water sublimation during the etching process. And we think this is because they're highly decorated with sugars and kind of hold on to their water very strongly. So freeze-etching allowed us to visualize all these structures that were previously pretty difficult to see or understand. And now we have an idea of how this kind of unique structure on the enterocytes actually functions. We were able to build a model of it actually. So here we have the microvilli with their links holding them together. And above that we have this dense network that we think serves as sort of a size exclusion filter in a way. And on top we have this termini that all are on the same plane and probably act as more of a protective function to protect the enterocyte below. All right, so let's move on to optimizing replicas. So first, let's start with some general guidelines. So first of all, most important thing when making any sample really is to start with something high quality. So this means that the sample was healthy beforehand, it did not degrade over time, and either it was fixed well before you froze it, or you can actually fast freeze. If you're going to fast freeze in the high pressure freezer, for example, it's best to kind of get that sample out of the animal or tissue culture, whatever is going on, and into the high pressure freezer as quickly as possible. For a lot of things, this can be done on the order of seconds if you're really good. And this will preserve everything the actual way it is. So you get the highest level of kind of temporal detail. Nothing is degraded yet. It's also really important to get good ice. So it has to be in vitreous ice. If there are ice crystals, they will look very poor, and it's really not even worth fracturing. And the second part is where a lot of problems often occur. So while we're sublimating water away in etching, we actually don't want a lot of water to get into the freeze fracture apparatus. So in general, it's good to avoid humidity with any cryo workflow, and this is especially the case for freeze fracture. And sometimes you can avoid and sometimes you can't. In Tennessee, it's a very humid environment. We run dehumidifiers to reduce the humidity as much as possible, but there is still some. The second part is not allowing essentially air into your freeze fracture apparatus when you insert the sample. So reducing that amount of contamination is very important. It'll allow you to reach higher vacuum quicker and cause less problems with your samples. And kind of along those lines too, it's very important to allow the freeze fracture apparatus to reach high vacuum before starting a fracture etch. This will prevent contamination of your samples from water. So oftentimes the cold sample will attract frost to it. And after you've done your fracture etch, if you get frost on your sample, it will be preserved in the replica and look quite poor. So it's important to avoid that. Kind of along those lines, it's really important that as soon as you're done fracturing or etching to actually form that replica without any delay. So as soon as that blade is off the sample, the metal evaporation should start. Now let's go on to metal shadowing. So the optimum angle and whether you have rotation or not varies greatly between samples. In general, the lower the angle of the metal shadow, you can get greater detail of your sample. But if you have a lot of topography in your sample, you're going to end up with large regions that aren't shadowed at all, and you will have difficulty analyzing those. Additionally, whether you do a rotary shadow or unidirectional shadow will greatly change how you're able to interpret different structures. For example, Ti-Junction strands are almost always unidirectional shadowed because it makes it easier to understand what the strand looks like. But freeze-etch are often rotary shadowed. In general, I like 45 degrees unidirectional shadow for freeze fracture, and for freeze etching, more like rotary shadow for sure, and maybe 20 degrees. It's something that you have to play around with a little bit to optimize. All right, so let's get into optimization of replicas for biological fidelity. So we're going to go into a technique I developed to preserve maximum amount of biological detail, but this is at the expense of contrast. And we'll kind of go through this. So what we're actually imaging when we're imaging these kind of samples, we're not imaging the biological surface itself, we're imaging the replica of the biological surface. And this is primarily the metal evaporation at a shadow that we're imaging. So if the metal does not accurately preserve the details of the biological surface, then we can't see it in the electron microscope. And typically we use platinum. Platinum is a very good metal for this because it forms very small grains, but it does still form grains. In fact, any pure metal that you deposit on a biological cold sample is going to form crystals. And we can actually see them. If you look carefully, you might be able to see some platinum lattice planes in this image. This is a very high magnification of the Ti-Junction strand. We do the Fourier Transform of this image. We can see these spatial frequencies, and we can actually characterize these spatial frequencies and find they correspond very well to platinum crystal planes. So the crystallization of the platinum is interfering with our ability to resolve biological details. We did this similarly with the luminal surface of the bladder epithelium. This has that uroplakin 2D crystal. That's a very useful tool for understanding resolution. Here we did a rotary shadow, and here's the uroplakin complex. See, they should all actually be completely uniform to one another, but we can see that the platinum did not deposit uniformly, and this is again due to crystallization. So we thought, what can we make replicas out of that would not crystallize? And there's one answer that's fairly obvious that we tried, which is just to use amorphous carbon. So in this case, we're just cutting out the platinum shadow completely. And we're actually shadowing a little bit with carbon instead, but mostly this is carbon from overhead. So we have the luminal surface of the bladder again, just as a resolution test. So these are all 2D crystals. And that allows us to use the Fourier space to kind of figure out how much biological fidelity we have. So over here we have freeze-etch, just rotary shadow platinum replica showing the surface. And here, this is the same replica 3 times using amorphous carbon instead, using different imaging techniques. So the platinum replica is kind of limited to about 3 nanometers resolution, and that's just a inherent property of the platinum crystals. When we look at the carbon, we don't have a lot of contrast. In fact, when we're near focus, since there's very little amplitude contrast, there's almost nothing to image at all. Although if we look in the Fourier space, we can see there's actually quite a bit of biological detail there. Spatial frequencies extend quite far. We can apply some phase contrast techniques, either using defocus, which is sort of the conventional way of adding some phase contrast in TEM, or we can use a phase plate and get some additional details out of these samples. And what we see is that we get spatial frequencies that extend quite a bit further, up to, I think in this case, it's 1.8 nanometer, but we've actually gotten smaller as well. We can take these spatial frequencies and actually filter on them and kind of get rid of the noise in the image. When we do that, we can see the platinum sample actually looks pretty nice, but only the central core of the uroplakin is really getting shadowed. Probably has something to do with the topography along with the crystallization of the platinum. When we look at the carbon, on the other hand, we're able to resolve a lot more details of this complex. However, there is very little contrast, so that's an issue using the carbon replicas. We thought it might be interesting to try and overcome this lack of contrast using kind of a single particle approach similar to what's done in cryo-TEM. And so in that case, we looked at connexin 26, which is a gap junction protein. And here we have just a freeze fracture carbon replica of connexin 26. And what we did is just picked all the particles in this one image and then did some alignment and averaging, and we were able to get this image out of it. Keep in mind, this is just this many particles here. And we're able to see this kind of star shape in the center of the connexin. and also some of the outside detail. Out here, this is sort of the carbon shadow, this dark region. When we fit that with the crystal structure of connexin 26, we find it actually fits quite well. Now, this technique is quite limited in replicas to use single particle because we only really have one view. You can only see them from the top down, so it's not like you can do a full 3D reconstruction. But it does have utility in understanding how some of these proteins fit into greater biological context. So we also went back to this Ti-Junction architecture with this new technique. And what we were interested in this time was not this interaction, which we felt was pretty well studied at that point, but instead whether this across-strand interaction was actually occurring. So no one has been able to kind of confirm this with any direct evidence. Mutations in this interface often cause the strands to fail to form, but that doesn't necessarily prove much. So we were interested if we could actually resolve that there was a space between these two transmembrane regions in the Ti-Junction strands. So we used carbon replicas for that. Well, here, first I'm going to show you platinum replica of a single Ti-Junction strand. So the strand is here, and we can see a lot of crystalline lattice planes from the platinum, but we can't see a double structure at all. It's just not present. And we think that's because the biological resolution of platinum replicas is just not good enough to resolve this small of a structure. So we used carbon replicas instead using some phase contrast techniques. And we were actually able to show that there is, looks like two lines in a single strand, and the lines are about 4 nanometers apart. And so that gave us a good amount of confidence that these Cloudin strands, which are also about 4 nanometers apart, are probably actually this double-stranded versus a single-stranded. We can actually resolve that there's probably a difference between what a single strand would look like versus two, and they all appear to be two. All right. With that, I just have some acknowledgements. So first, I would like to acknowledge my postdoc advisor, Bashar Kashar, and all my co-authors that were at NIDCD when we published those studies. And then I would like to acknowledge Vanderbilt University and Cell Imaging Shared Resource Core Facility, where I work now, the management of the Cell Imaging Shared Resource, Matt Tiska, Jenny Schaefer, and Jay Jerome, as well as the people I work with every day, Maria, Rachel, and Mary. With that, I will hand it over to Seth.
Thank you, Evan. My name is Seth Villarreal. I'll be talking briefly on the ACE900, which is an automated freeze fracture system that can assist in the type of workflows that Evan just presented on. The high vacuum workflows that can benefit research goals involving freeze fracture and freeze-etch are generally fairly complex machines. The ACE900 is a large tabletop instrument with the ability to load samples under vacuum on a cryo stage and then carry out freeze fracture and freeze-etch in a controlled, dedicated environment. The loading interface allows samples to be picked up either through a manual system or through the VCT 500 shuttle, allowing the sample to be worked with in a variety of different ways. Keeping a sample preserved throughout the entire workflow is a critical part in maintaining its sample integrity. The load block system on the ACE900 has a couple of different options. On the left of the image was the docking port for the VCT 500. On the right was for the manual transfer rod. Either of these allow a sample to be loaded onto the cryo stage of the ACE900, letting you work in a vacuum up to 10 to the -8 millibar. And the cryo stage can operate at a temperature down to negative 185 C. With the option of 2 E-beam emitters, the cryo stage can tilt, rotate, and rock during coating, allowing the sample to be prepared for any workflow needed. The freeze fracture and coating can be done either through a manual or automated fracture, and even fracture into the E-beam, and the knife for freeze fracture can be exchanged under vacuum, such that multiple samples can be handled at once and maintained in that very controlled environment. During evaporation, the coating rate is adjustable, and it's possible to have separate E-beams for platinum carbon coating and pure carbon coating. Furthermore, working with the samples, there is an option for camera or direct viewing, provide as much flexibility as possible in trying to handle these delicate workflows. The ACE900 is one of the machines and Leica's dedicated instrument for carrying out freeze fracture and freeze etch. Thank you, Evan, for the chance to share this particular device and to hear about your work. At this point, we'll be pausing to answer any questions. Thank you, everyone.
Thank you, Evan and Seth, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Our first question is, are there ways to mark proteins or complexes of interest in preparation for freeze fracture?
I think I can answer that. This is Evan. So one way of marking structures in freeze fracture, freeze etch is to do Immunogold labeling. This is a rather tricky technique to do. John Rash has some excellent papers describing the technique. But essentially, after the replica has been formed in the freeze fracture apparatus, you would digest away the tissue with something like SDS rather than bleach in an attempt to keep some of the membrane protein still attached to the replica. And then you can Immunogold label those attached proteins and then kind of go in a more or less standard workflow in the TEM. And in that way, you have whatever you want to look at marked with the gold. You could probably also do this with something like a quantum dot and do some correlative microscopy with it.
Great, thank you so much, Evan. It looks like we've got another question for you, and this one is 2 parts. First part, is there any experimental demonstration that etching does not facilitate some collapse of fine fibrillar elements? And the second portion is another way to ask is this: has FFTEM glycocalyx been shown to have the same appearance of the fibrils as hydrated atomic force microscopy?
So that's a really good question. So I don't have a definitive answer on this, but I'll give you a couple thoughts I have on it. So we have looked at various etchings on the glycocalyx, and if you over etch, so this is just remove too much water, the glycocalyx does collapse. So we were trying to minimize the etching to kind of get a single layer of mucins that are still kind of supported by vitreous ice on the back for this study. I think it would be really interesting to look at it through AFM. I don't know how you would image from the side like that. Have to think about a way to kind of section appropriately, but you would definitely look from the top and look at the termini at the very least.
Thanks, Evan. Our next question: how delicate are the replicas, and can etched samples be stored or transported?
So the replicas, when they're being floated on the bleach to remove the tissue, are extremely delicate. This is when you can easily lose the sample. So the thicker the replica, the more likely it is to hold together, but also you'll get less details. Once you've got them free of their tissue and kind of collected them onto EM grids, they are very robust and stable. You can keep them in a grid box for years and they'll be fine.
And our next question, instead of pure metal, have you been using any other types of metals?
Well, we have explored this quite a bit. So the ideal shadowing material would be something with a high Z, so that it scatters electrons through amplitude contrast, and would also kind of accurately replicate the biological surface. So this would kind of just sit where it's deposited and not form a crystal. Now, we found that basically any pure metal is going to crystallize under these conditions when deposited. So, and this is especially known for things like gold, which are routinely used for SEM. But it is possible that a mixture of metals to form an alloy might be very suitable for this. And maybe some of the lighter metals may work quite well as well. But then you're back to phase contrast, and you might as well just be using carbon at that point.
Great, thank you so much, Evan. So we do have another question that came in. It's a little bit of a long one, so bear with me here. The formation of crystalline PTC replicas seem to be an unusual feature of the ACE 900, which is, of course, not desirable. This is not observed in other freeze fracture machines. Have you studied the techniques of John Rash to get a high level of detail from the PTC freeze fracture replicas? And could these methods or hardware modification be implemented in your hardware?
I guess I can start with this and then hand it off to Seth. So the crystalline formation of platinum is inherent in all platinum carbon replicas. It's just not normally imaged because you need a very high magnification to see it. Most of those replicas were actually developed or made on a Balzer type freeze fracture machine, so probably closer to something John Rash is using. I know he gets very good replicas by having very, very good vacuum and depositing much colder, and this will definitely produce better results. But you will get crystals no matter what you do if you're depositing a pure metal. And I'll hand it off to Seth for any additional details here.
Seth, do we still have you? It looks like we may have, unfortunately, lost Seth, but, you know, unfortunately with technical difficulties, what we can do, though, is for those of you that have asked that question, what we will do is we'll have Seth reply. We will have him reply via email to you. So we will be able to answer, you know, the rest of your question off of what Evan has already answered. So we apologize for those technical difficulties. So let's go to here. We've got another question. So Evan, I believe this one will be for you. How can somebody tell if a freeze fracture knife needs to be replaced?
Well, you will be able to tell because your replica will start to have marks in it from the knife. So if you see unnatural kind of indentations in the replica and maybe a cell or something like that, it's a good indication that the knife needs to be changed.
Thank you so much, Evan. And it looks like we may have time for one more question. Why do you refer to why do you refer to pure metal when it is a PTC shadow?
Okay, that is absolutely correct that it is a platinum carbon deposition. So the E-beam actually has a carbon crucible in it, which has a piece of platinum in it, and that's what deposits onto the sample. So I think it's something like 5% carbon, 95% platinum. And the idea there is that the carbon works to interfere with the platinum crystallization, kind of like how steel works. And so that's the correct terminology. We just say pure metal as kind of a general terminology instead of like a mix of different metals like an alloy.
Great. Thank you so much, Evan. So we want to thank Evan and Seth for their presentations today. Fortunately, since we did lose Seth, Evan, do you have any final comments for our audience?
Freeze fracture and freeze etching are useful techniques, but they take a little bit of skill to learn. So if you want to do these techniques, patience is required.
Duly noted. Thank you, Evan. Thank you again, Evan and Seth, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information that you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Until next time. Bye-bye, everyone.